Well, let's talk about the genetic errors within what you've explained so far, why that doesn't work.
Okay. So the first thing they say that each gene codes for one protein. And so let's start there. So then they do the Human Genome Project. And by the way, I didn't know this until fairly recently, but I thought they took a person, extracted the DNA, sequenced it like Sanger sequencing, which is just a way of, ATGC all the way down the line. And that was the sequence of that particular human being. But that never happened. What they do is they take multiple short segments from many, many people. They chop them up and then they send it to the computer to align them. So it's like a sentence and there's overlapping segments, and they create rules to tell the computer how to do it. And then it comes out with a hypothetical sequence that's a mixture of these literally hundreds of people. Now, they say that there's at least 5 to 10 or maybe 15% that's missing. But when you read that, you think about it, if you've never had the whole strand, how do you know what percentage is missing? It's like the analogy I use, like a book. So you have a book, and then somebody gives you 56 million words, and they say, arrange this into a book. And you say, which book? Well, I'm not going to tell you. You just arrange it into a book. So you come out with certain rules, but there's no way to know whether that's the right book, because how would you know that? So anyways, that's an interesting thing. So they don't really know the human genome. It's never been done, or any other living being. It's never been done. So, but anyway, so they have these genes, and then they say, well, we found 10 to 20,000 genes in this hypothetical genome. And then they do the number of proteins. One gene codes for one protein. And there's somewhere between 100 and 200,000 proteins. And my usual statement now is, apparently geneticists are not good at arithmetic, because that means there's even somewhere between 80 and 180,000 missing codes for proteins. So what's the code for those other 180,000? They don't know. Anyways, so that's a problem, right? Because the whole thing doesn't add up. Now, the second problem is they say that the DNA is wound up into chromosomes, right? You've seen chromosomes. Humans have 23 pairs of chromosomes, and they're all the same, 22 somatic ones, an X and an X, or an X and a Y. And they say that the DNA is wound up, packed into those chromosomes. And that's so it can divide and form two, and then it has to unwind so that it can be read, right? You have to open the book. So I happen to have been, and when I was an undergraduate at Duke, I was allowed into this graduate level biology class, cell biology, by a guy named Nichols, who was teacher of the year at Duke 3 times, great teacher, nominated for a Nobel Prize for discovering histones, which he said are the proteins that fold the DNA into a package to go into the chromosomes. Okay, so he says, I'll show you how it works. So he sends one day, go to class, and there's stanchions all around the perimeter, and he wounds a yarn all around 9 times. And he said, this is approximately the length of the DNA. And then he had a thimble, like you use with sewing, this big. And he said, that's the size of the chromosomes. So he starts winding the string into the thimble, and he gets about halfway around one turn, and he can't put any more. He got a stick and stuffing it in, and he goes about 3/4 away, and then he can't get any more. And he said, and isn't it amazing that life can do that? And everybody stood up and cheered, except me. And I'm sitting there thinking, wait a minute. He just showed us that DNA can't possibly go into that thimble. So I don't know what the chromosome is, but I have serious doubts as to whether it's wound up DNA. So that's the next problem. Then the DNA is made into mRNA in the nucleus. And then it goes to the cytoplasm. And we know that the nucleus has a membrane. You can see it on a light microscope. We also know that the pH, right, you can measure that with a probe, of the cytoplasm, the watery part, is different than the pH of the nucleus every single time. So that means, according to normal theory, the hydrogen ions, that's the part that determines the pH, can't equilibrate across the membrane, right? So there's a different concentration in the cytoplasm versus the nucleus, which is why they have a different pH. Now, here's the interesting thing. mRNA is approximately 1 million times bigger than a hydrogen ion, according to their way of thinking. So if the hydrogen ions can't go back and forth, how does a mRNA get out of the nucleus through the membrane without letting the hydrogen ion in to equilibrate? And they say, well, that's through the nuclear pore. It's a hole. Well, if there's a hole big enough for an elephant, why can't the mosquitoes go back and forth? Anyways, they don't say. So then they made up an endoplasmic reticulum, which is a tube that attaches, and then it's got a whirligig on it, but they can't show you a picture of the whirligig. So the whole thing is just sort of nuts. And then it goes to the ribosomes, and it gets translated, made from mRNA into protein. That's what they say. So I went looking to see if there's actually ribosomes. And Harold Hillman, who's the best biologist who ever lived, according to me, not just me, but he did a 100-page treatise on ribosomes. And what he said was you can never see them in a light microscope. They're too small. You can see them only in an electron microscope, which means you froze it, stained it, dehydrated it, embedded it in a resin, put heavy metals on it, etc. And every picture is a perfect circle of a ribosome. And you also had to blend the tissue in order to do this. If it's a circle on a picture, it was a sphere in real life. And so he asked the question, what are the chances if you ground up an orange and froze it and dehydrated it and stained it and embedded in a resin, that every piece of the orange would be a perfect circle? And the answer is 0. There's no chance. So then he said, okay, I'm going to take tissue that can't possibly have ribosomes, by everybody's account, and I'm going to do the same electron microscopy steps, and I get these perfect circles, which means it's an artifact, which means it's a gas bubble, which we know are formed when tissue dies and you fix it like they do. So there is no evidence of a ribosome. Just one more word about that. The word ribosome means rib, right? Rib, o, or of, the soma, meaning body. And when I first saw that, I thought, where have I heard that life was created from the rib of the body? I mean, that's what it says in the Bible. The woman, I think, was created from the rib of Adam, of the body of Adam. So I think that ribosome story was mockery. So proteins are what we're made of, allegedly. So they made how the proteins are made, it's the rib of Adam. Ha ha. So there's a lot more, but that should get us started. The whole thing is basically hooey.
Well, as we start to pick apart the current theory, I think it's important we put out there the fact that you're agreeing that there are genetic changes, where the difference is conventional thinking is making them the cause, versus you're saying there's something else upstream happening.
Well, I wouldn't, just to clarify, I would not use the word genetic changes. There are changes in the chemical that they're referring to as DNA. There are also changes in the amount or appearance of the chromosomes. In fact, I know people well who go around saying that the cause of cancer is aneuploidy, which means all normal cells have 23 chromosomes, 22 somatic ones, X and X, X or Y. But if you show a tissue slide to a pathologist and you say, is this cancer or not? They look at the chromosomes. And if there's the 23, then they say, no, it's not. But if it's abnormal, like 22 broken ones or 28 or 100 all mixed up, that's cancer. And the more aneuploid it is, in other words, the more different from 23, the more aggressive the cancer is. So there's no doubt that an effect, just like you were saying, of whatever started this process ends up with, here's a scientific word for you, wonky chromosomes. They're not right. And wonky pieces of this chemical you're calling DNA, right? That's not genetics. That's just there's wonky chemicals because something is wrong. And so you get stuff. It's like you blew up the building and you get broken armchairs. The armchairs didn't cause the blowing up of the building.
We can call it what we want to call it, but whether it be genetics, DNA, it's important too to put this out front that when we look within a body at these changes in different cancer cells, it's actually different changes and between two people with the same kind of cancer.
Yes.
So it's not like we're seeing the same change across the board within a body or between bodies. And we can use any example, prostate cancer, breast cancer.
Yeah. It looks like somebody put dynamite in your house and everybody's house will have different books and different chairs and different whatevers. There's commonalities, like everybody has a kitchen, so you get, blown up pots and pans in most houses, but they're not the same. And by the way, I would say it is now proven fact that every tissue in the body has different kind of DNA, which is a real problem. If the DNA is the code, it's got to be the same everywhere. It's your code. That's what we're told. But if your liver has a different set of DNA than your spleen does, like that's a problem because that's not supposed to be how it works. But that is the facts. And it changes through your life. So you don't have the same DNA as you do when you're 5 as when you're 60. And so that also puts a huge crimp in that theory because that's not supposed to happen. You're supposed to have the same DNA, supposed to be normal, except if you get a mutation in an oncogene, and then you got this cancer-causing activator or suppressor function, and then you get excessive cell growth. That's what they say.
To put another crinkle in things before we move into how you see cancer developing, let's talk about the experiments where they take the nucleus and the cytoplasm of normal cells and cancer cells, and they start moving that nucleus around, and that's going to lead to what you see as the true origin of cancer.
Well, and I have references in my cancer book, and Thomas Seyfried has worked on this a lot. So if you have let's just say there's cells, even that I would say is questionable, but let's say there is for now. So you got this cell and it's got a watery part, cytoplasm, which is like organized gel phase water. Everybody would agree with that. And then you got a nucleus in the middle, or not exactly the middle, but it's inside there. The DNA is in the nucleus, right? And so if you take a cancer cell and a healthy cell, and you take the nucleus out of the cancer cell and put it into healthy cell cytoplasm, you don't get cancer. Whereas if you put a healthy nucleus into a diseased cancerous cytoplasm, the progeny are cancerous. What that tells you is the location of the error is in the cytoplasm, not the nucleus. And since the nucleus is where the chromosomes for the aneuploidy theory or the DNA for the oncogene theory reside, it's not there. That's not the location of the problem. And this has been repeated and referenced. And, what's so frustrating is, like, nobody responds to this. Like, that's a problem for the theory. And so you would think there would be some explanation for how this observation, this experiment that disproved your theory goes unquestioned. They just ignore it.
Before we move into the cytoplasm and get into how you see cancer starting, any other crinkles in the conventional thinking, big ones that we need to point out?
I mean, one of the things to think about is if you have a theory and you apply a remedy based on a theory, it should work. And, the Australian government, for an example, looked at the positive benefit of chemotherapy across the board. I think it was 2003 or so. So this is 30 years after the war on cancer and improvement in the whole thing, allegedly. And they said there's between a 2.5% and 3% improvement in the outcomes based on chemotherapy, which is like nothing. I mean, if you're going to go to a business person and invest $10 trillion and you make the same toaster at the end, like, that's a problem because you got a bad return on your money. So that's one problem. And the fact that they still use the same chemotherapy drugs as 1970. I mean, they're cleaned up a little bit and there's some new ones, but it's really gotten us nowhere. And as you pointed out, these changes that they're seeing are downstream. And nobody has ever been able to change a gene, put it into a mouse, and form a new cancer. They can't do that. They can put a bunch of stuff in there, and poison the mouse, but to just take the gene, so that's what they're saying is the problem. You have to prove that. And so you have to do an experiment. You take that gene, put it into an animal, see if they get cancer, and then you put other genes that don't cause cancer, allegedly, just to see if it's not the process of putting a gene in a mouse, right? That's called science. Science is you do everything you can to disprove your theory. Every control experiment, well, what if it's just sticking needles in mice? Well, some do and some don't, so it's not that. Or maybe that is what it is. So and they've never done that. They've never proven the theory. Like, that's a problem, folks. If you're going to say a theory, you got to prove it in science. Otherwise, unless you can observe it with your eyes, then, I know that this is nail clippers. I can ask you if it is. That's another way of knowing things. But this one, you don't see it. There's no observation, no sensory experience. You don't really know, so you have to do science.
If you enjoyed that clip, you're going to want to head over here and catch the full episode. I'll see you over there.
I don't know the amount of money spent on researching this oncogene theory, and they got nothing. Nixon announced the war on cancer. We're going to spend whatever it takes, and in 10 years, cancer will not